Next topic is very interesting topic to the microbiology students, and this is the last topic of my portions. Biosynthesis of gram-positive and gram-negative cell wall. You know very well. Okay, the cell wall is the exterior component of the bacterial cell, and it surrounds the cell membrane, and this is a protective layer of the bacteria. So that protective layer, the peptidoglycan layer, is the very complex, and we are going to see the biochemical, the composition of peptidoglycan layer. Usually, the peptidoglycan layer is made up of two sugars. Okay, that is N-acetyl muramic acid, N-acetyl glucosamine. It is represented as G and M, N-acetyl glucosamine and N-acetyl muramic acid. You can see here the lengthy chain of NAM, NAG, NAM, NAG, NAM, NAG. So this is NAG and this is NAM. Okay, this is going to be continued, and this bond between these two sugars are called as beta 1,4 linkage. So this linkage is beta 1,4 linkage, and if it is like this one, okay, that is alpha, and if it is upper to lower, okay, that is called as beta, because it is always the hydroxyl moiety is always there on the top. Okay, that is there, it's called as beta moiety. So beta 1,4, this carbon is 1 and this carbon is fourth moiety of the muramic acid, beta 1,4 linkage. And this 1, 2 and 3, the third carbon atom having a lengthy chain of 4 peptides, that is tetrapeptides. So the four different amino acids, L-alanine, D-glutamate, and L-R3. R3 means the different, maybe the different compositions may be there, and D-alanine. And this is the composition, okay, maybe in the third position, that is the CH3, CH, and CONH, and attached with, okay, different types of 4 different amino acids. This third amino acid may vary from one organism to another. So we can go to the detailed way by explanation. The disaccharide peptide unit and peptidoglycan, okay, the glycan backbone consists of alternate residues of N-acetyl glucosamine, that is G and M, N-acetyl muramic acid by beta 1,4 linkage. The carboxyl group of the lactyl moiety of the N-acetyl muramic acid, that is why I told, okay, this is a carboxyl moiety of N-acetyl muramic acid, consists of tetrapeptide, and this tetrapeptide is usually L-alanine, D-glutamic acid, L-R3, residue 3, and amino acid varies with the species, and finally D-alanine. So D-alanine will be in the last. So this may vary from species to species. Okay. The peptide linkages are all except for the between, okay, there is a D-glutamic acid and the amino acid in the position 3, which is linked, directly linked. Okay, there is gamma linked. Okay, this one. And the third one is the different. And alpha carbonyl of glutamic acid can be free and amide or substituted. Okay, that may be either, that may be in the free form, or otherwise that may be a substituted form. Okay, that may be there. Next, coming to the different types of peptidoglycan cross-linking. Okay, this is one chain, NAM, NAG, NAM, NAG, NAM, NAG, and this is NAM, NAG, and this is also another type, okay, B type, and this is also NAM, NAG. This is total the interlinking of amino acids. The first we can see one by one, the A. A means a direct cross-link between diaminopimelic acid. So this is direct cross-linking between the diaminopimelic acid or pimelate, and it's called as, in short, it's called as a DAP, diaminopimelate. Okay, that is present in the bacteria, and pimelic acid and D-alanine acid occurs in the gram-negative bacteria. So this is alanine and this is pimelic acid. This is going to be attached by the NHCO bond. Okay. Second one is an amino acid bridge between the two tetrapeptides. Okay, that is, and this is amino acid bridge between the two tetrapeptides. Okay, that is in many of the gram-positive bacteria. So this is the C-terminal alanine moiety, C-terminal D-alanine moiety of the. And next one is carbonyl moiety. This amino group and carbonyl moiety are going to be reacting with an amino acid bridge. It is like a peptide bridge. Okay, and third one is the schematic diagram of drawing of cross-linked peptidoglycan. This is a completely cross-linked, and tetrapeptides are indicated by straight lines. Okay, these are all the tetrapeptides are linked as straight lines, and the glycan chains in some bacteria have been found in certain 20 to 100 sugar residues. Okay, that is that is some of the bacteria may have 20 to 100 sugar residues also may be present on the cell wall. That is the glycan chain of the cell wall. This is a major component that may form the glycocalyx. So glycocalyx is very important to defend some of the macrophage maybe engulfment, or otherwise by the action of some any of the antibiotics or extreme environment, anything. So the glycocalyx is a protective layer, okay, next to that cell wall. Next is synthesis, how these two are synthesized and what is the metabolic pathway involved in this. The major precursor is fructose 6 phosphate, and this fructose 6 phosphate is converted into glucosamine 6 phosphate by donation of one amino group. You can see here this is a first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. This is a fructose moiety. Usually the fructose moiety having a CO in the second carbon. You can see here the second carbon CO is converted to and CnH2, that is glucose amine group. This is amine group donated by glutamine and converted into glutamate, that is glutamic acid. In the second reaction, acetyl coenzyme A is going to be reacting with the glucosamine. This acetyl moiety, CH3CO moiety, is going to be attached, okay, in the second carbon. Already second carbon having NH2. So to the NH2, now we can, this is open chain arrangement, and this is the Haworth's projection formula. This is a pyranose form, and in the pyranose form this is the first carbon and this is second carbon. You can see the second carbon is having NH2, this NH2, this NH, and CH3CO is going to be attached. So this CH3CO is coming from acetyl coenzyme A, and the coenzyme A is liberated in the form of COSH. So now the product is named as N-acetyl glucosamine 6 phosphate. This is N-acetyl glucosamine. Okay. N means the acetyl moiety is attached to the N moiety, that is nitrogen. That is why N-acetyl glucose, this is glucose, and amine with NH2 moiety. 6 phosphate, with the 6 phosphate, 6 carbon, 1, 2, 3, 4, 5, and this is the 6 carbon with the phosphate. Okay, already 6 phosphate is there, fructose 6 phosphate and glucosamine 6 phosphate and N-acetyl glucosamine 6 phosphate. And the third reaction, N-acetyl glucosamine 1 phosphate is formed. Instead of 6 phosphate, it's a conversion to 6 phosphate to 1 phosphate. And again, UTP2 PPA will be formed, and UDP N-acetyl glucosamine is formed. Okay, this 1 phosphate is going to be removed. And this is going to be removed, and at the same time, this will be combined with UMP. That is why it is called as UMP with phosphate. Okay, this phosphate is going to be attached with UMP, and the incoming molecule is UTP, uridine triphosphate. So uridine triphosphate are coming, and 2 phosphates are removed. Remaining 2 removal of 2 phosphate, that will be uridine monophosphate. This uridine monophosphate is going to be attached with the phosphate, and this is called as UDP N-acetyl glucosamine. So this UDP N-acetyl glucosamine is converted into UDP N-acetyl glucosamine and 3-enoyl pyruvyl ether. So 3-enoyl means third carbon, first, second, third carbon. Now this carbon is going to be involved. So by this reaction, okay, number 5, and phosphoenol pyruvate is converted to phosphate. So the phosphate is removed from phosphoenol moiety, and the phosphoenol moiety is going to be attached. The phosphate is removed and enol moiety is going to be attached. So this is the enol moiety, CH2 and double bond C, COOH to the third carbon. So that is what is called as UDP N-acetyl glucosamine and 3-enoyl pyruvyl ether. Pyruvyl ether is, okay, this one. Next continuation of, okay, this one, sixth reaction is dehydrogenase reaction, and NADPH plus H plus involved in this. Okay, to remove, to add, okay, 2 hydrogens to pyruvyl, enoyl pyruvate, that is CH2, CH, COH will be formed, and this is called as UDP N-acetyl muramic acid. So previously we had UDP N-acetyl glucosamine, so this is already formed, and from glucosamine muramic acid is formed, and now we have to attach 4 different peptides. So this peptide is ready, L-alanine, D-glutamate, and LR3 and D-alanine and D-alanine moiety. Okay, that is with the 4 ATP molecules are involved, and this will be attached with the carbonyl moiety. Okay, this is third carbon containing enoyl moiety, and this enoyl moiety having the carboxyl moiety. To this carboxyl moiety, all the peptides will be attached. So this will be 1, 2, 3, 4 peptides. So this is not a tetrapeptide, this is a pentapeptide. and ultimately this will be active one, and this will be going and fusing with another chain of reactions, and that will be removing one alanine moiety. By this way, all the amino acids are combined together, all the four different amino acids are combined together to the carbonyl moiety of the third carbon atom of uridyl N acetylmuramic acid, and now the UDP muramyl pentapeptide will be formed. This is the major precursor for the synthesis of peptidoglycan layer. So with this, we can come to the end of the session, and if you have any doubts, you can clarify and you can raise the questions. Thank you.